Okay, hello everyone. Hope you can hear me. I was getting worried there for a second that I couldn't get into the session, but here we are. So let's continue with some interesting talks on pollination. We've got 3 more talks to go, and we start off with Sam on Erikas. And Sam, it's immediately over to you. I want to try and stick to the time.
Okay.
So if we can try and share, see if we can get that up.
Can you see it?
Great. Yes, we can see your presentation. Over to you.
Okay. Hi, I'm Sam McCarran, and I'm really excited to share my recent research with you, which is sending private messages about the role of UV in pollination of Erika. And my supervisors are Jeremy Midgley, Anina Kutsia, and Steve Johnson. Let's start with a little bit of background. Pollinators have different visual abilities and they react differently to flower colors. This here is a great example. So the 4 flowers on the left are pollinated by bees, and the 4 flowers on the right are pollinated by birds. Some of you might have already noticed that the 4 bird-pollinated flowers are all red. And this is a general trend. Bird-pollinated flowers are more often red. And one could assume that this might be due to birds having a preference for red, but that is not the case. And it has been found that bees have a low discrimination ability between red and green. So those red flowers actually avoid bees who would rob their nectar while still being very visible to their bird pollinators. If we now have a look at UV, UV ranges from 300 to 400 nanometer wavelength, and it is not visible to us humans, but some pollinators can see it. There have been very few studies on bird pollination in regards to UV, because most hummingbirds cannot see it, but there are some recent studies that show sunbirds should have the genes to be able to see UV. And in general, UV radiation is harmful also to plants. So most plants have compounds that absorb UV and protect them from the radiation. So reflecting UV should really only evolve if there is some benefit to it. And those benefits could be that the flowers could be more conspicuous to their pollinators, but they could also avoid detection by undesirable visitors, just like the red flowers avoiding bees. So I set out to quantify UV for different pollinator groups, and then to test pollinator preference in the groups where I found UV. I chose the genus Erika for this because Erika has undergone an extreme radiation in the Cape. There are almost 700 different species of Erika here nowadays, and they have a diversity of flower morphologies. They grow in different habitats, and most importantly, they have a number of different pollinators. So part of their radiation might also have been driven by the pollinators. I sampled a total of 125 species and covered 5 different pollination syndromes. And I just want to introduce you to all of them. Number 1 is a long proboscid fly pollinated Erika. Those are typically light pink in color. They have long tubes with a narrow opening and flaring lobes. Number 2 is other insect pollinated Erikas, which are most often small and pink. But when Rebello and al. Classified the different pollination syndromes of Erika, they merged quite a heterogeneous group together. So presumably most of those Erikas are pollinated by bees and other hymenopterans, but there are also some other pollinators, like, for example, moths. Number 3 is a bird-pollinated Erika. So it's pollinated by a sunbird, and they also have long tubes, a slightly larger opening to allow for the bird beak to be inserted, and they come in a wide range of different colors: red, white, pink, green, yellow. Some are even two-colored. Number 4 is a rodent-pollinated Erika, and then number 5 is a wind-pollinated Erika. And this photo was actually taken under a microscope because the flowers are so tiny. The ones in the picture were only about 1 millimeter long. So when I was looking at my reflectance spectra, I chose a 10% contribution of the UV to the total brightness of the flower as a cutoff. And you can see this cutoff as the red horizontal line in this figure. So let's jump right into it and have a look at the rodent and wind-pollinated species. And for those, I found no UV, which for the wind-pollinated species is what I expected, because wind doesn't have any sensory systems, so shouldn't be selecting for any special colors. And the rodents that pollinate Erikas are nocturnal, so they should be more attracted by scent than by colors. Now, if we have a look at the insect pollinated group, there are very few species that reflect UV, and as I said before, presumably most of the species are pollinated by bees or other hymenopterans, and it has been shown that bees have a low detection ability of UV. So this is also what I expected. And 1% of the species I sampled reflected UV, which is 2 species, Erika hematocodon and Erika denticulata. And for Erika hematocodon, we don't know the pollinator, but it is quite interesting because it has, with its dark red flowers, quite a special flower color, and it would be really interesting to know what pollinates them. And then Erika denticulata is pollinated by moths, and moths can see UV. Now, if we look at the bird-pollinated species, I found that 17% of the ones I sampled reflected UV. So I went ahead and did a preference experiment with sunbirds. And I made those artificial inflorescences, which you can see the sunbirds visited quite happily. And I painted the flowers in red, and some with additional UV and others without UV. So the flowers looked exactly the same to the human eye and only differed in the UV range. Now, please have a look at the graph on the right. In the beginning of my experiment, I'm sorry, I forgot to mention, the flowers that had UV reflection, I filled with sugar water, so they had a reward. And the flowers that had no UV reflection, I left empty. So at the beginning of the experiment, the sunbirds had no preference. They visited the flowers indiscriminately. And then over time, the sunbirds learned that only the flowers with UV had a reward, and they started preferring them over the unrewarding flowers. So we can say sunbirds don't have an innate preference for or against UV, but they can see UV and they can learn from it and make foraging decisions based on this cue. Now, the last group is the long proboscid fly pollinated Erikas, and every single species I sampled reflects UV. And I also was able to take photographs of 3 of the species with a UV sensitive camera. So the purple areas are the UV reflecting areas, and you can see they reflect UV quite strongly. And have a look at the corolla opening. There is some kind of pattern which resembles a bull's eye. So this might be some sort of nectar guide to help the fly insert their long proboscis. So for the long proboscid flies, I also did a preference experiment, and I chose Erika aristata, which you can see in the picture being visited by a fly. I had 3 different treatments. One where I removed UV by applying sunscreen to the flowers. Then I had an open control that I left completely unmanipulated. And I had a scent control where I applied sunscreen only to the base and the pedicel of the flowers. So if the scent of the sunscreen would have any impact on the flies, we would be able to pick that up. And when I counted the seed set, it was quite a clear result that the scent control and the unmanipulated flowers, which both reflected UV, had a normal seed set, and then the treatment where I had removed UV had very low seed set. This indicates that the long proboscid flies have a very strong preference for UV in the flowers they visit, and they might even rely on the UV patterns to be able to insert their proboscis. And this is a result that I didn't only find for Erikas, but I also sampled some other long proboscid fly pollinated species in the Cape and found UV reflection in them. So here, 4 examples, which are Brachysiphon acutus, Gladiolus debilis, Pelargonium cuculatum, and Tritonia cooperi. So in conclusion, UV wasn't important for insect, rodent, or wind-pollinated Erikas, and it only played a minor role for bird pollination. Potentially, UV is more important for sunbirds for their sexual selection because sunbirds have plumage that reflects UV. Otherwise, UV seems to be not any more important than any of the other flower colors in sunbird pollination, but nevertheless, it might help the sunbirds to discriminate between co-occurring species and thereby reinforce floral constancy, and the flowers might also use it to avoid nectar robbers. But UV is very important for long proboscid fly pollination, and the preference of the flies for UV might have contributed to the evolution of this signal. Thank you to my supervisors and all the other people who helped me along the way, and special thanks to the NRF for funding me. And I'm ready for your questions.
Great. Thanks, Sam, for a lovely presentation. And it's I like how you took a whole genus, just like the genus Erika, and look at all of those species. That's really nice. Yeah. Nice to see. Let me just see if we've got questions coming in. Nope. So please, everyone, you're welcome to post your questions in the Q&A. Yeah, it's all just feedback. It's useful seeing that most of the presentations are student presentations. It's useful if you ask something or post something. So, Sam, I've done quite a bit of bird pollination work over the years. What do you? It's probably not a fair question, but in terms of Erika, I think you said 17% of species have UV.
Yes.
Do you think that's representative of bird pollinated species in the Cape in general? Do you have any idea, or do you think Erika is an exception, seeing that it's got long-tongued fly species with UV?
So I did sample a few species that were not Erika, but okay. I'm really not confident to make an assumption about species in general. I did find some species that are not Erika and bird pollinated and reflect UV, but I think for example, Saltera sarcocolor, I assume, is bird pollinated. Yes, yeah. But I think there are also phylogenetic constraints. So, for example, Saltera is very closely related to Brachysiphon, and Brachysiphon reflects UV. So maybe it just kept the UV reflection without actually being extremely valuable to bird pollination.
Yeah, there's no major cost for having UV. Yeah, then it might be remaining. Okay, so let's just check the questions in the chat. Q&A. There we go. From Glynis, are UV influence and scent correlated in any way, positively or negatively?
I'm not. I don't understand the question.
So bird pollinated flowers, as you said, have no scent. So I suppose what's happening is those scent,
okay.
UV, and because you exclude the scent of in your treatment. So is there anything with scent and UV?
Both sunbird and long proboscis fly pollinated Erikas don't have scent as far as I know. But I didn't measure any scent. Yeah, I don't know if it could be correlated, but I would be surprised. So the Erika denticulata that I mentioned before, that is moth pollinated, it has like a strong scent in the evening to attract the moths, and it still reflects UV.
I don't think. Yeah, just off the cuff, I wouldn't expect there to be some sort of correlation. It's just one trait of many that attracts a pollinator. Well, okay, another question. While absorption and containment of UV by plants is a protective measure, is reflection not protective too?
Yeah, so I also had a hard time wrapping my head around this. But for me, it was the best way to understand how UV damage works is like if you look at your own human skin, because human skin reflects UV, and through the reflection, the energy is absorbed into your skin and then moved out. And by that happening, there can be breaks in your DNA. And if you put sunscreen on, that absorbs the UV and just transforms the light energy into warmth. So that is the way how to protect yourself from damages.
Okay, nice explanation. We need to wrap it up. Let me just see if there's a quick one. If there's a comment. There's one more question. Are there UV reflective guides inside flowers? That's interesting. Inside flowers.
For the Erikas, none of the UV. So the nectar guides in the Erikas were UV absorbing, and so the whole Erika was reflecting UV, and then only the central part around the corolla opening absorbed UV. And then in other species like the Gladiolus that I showed, they are open because Erikas are closed, so there's no way for the pollinator to have a look inside. But in the Gladiolus, you saw there were darker red marks, and they also absorb UV.
Okay, great. Thank you very much, Sam. Interesting study. Thanks for the presentation.
Thank you.
Let's move on to the next presentation. So we move over to honeybush tea. Sure many of you heard about honeybush tea, Cyclopia. And Kirsten, are you there?
Yeah.
There we go. Okay. Just try and share your presentation.
Okay. Can you see it?
It's coming. Yes, there we go. Great. Over to you, Kirsten.
Okay, so good afternoon, everyone. My name is Kirsten Shaw, and I've just started my second year of Masters at the Cape Peninsula University of Technology. And the title of my thesis is Quantifying Pollen Transfer Between Cultivated and Wild Cyclopia Species in South Africa. So honeybush is an endemic genus to the fynbos biome, and it has great value in the tea industry of South Africa. And with the expansion of this industry, honeybush farms are extremely important to prevent the overexploitation of this resource. And with this comes a supply shift from wild harvested material to cultivated biomass. And because honeybush is endemic, it's very important to consider how best to develop biodiversity-friendly farming practices. And with honeybush being planted in its native range, there's a very high likelihood of wild populations growing in close proximity to where honeybush farms are located. And this opens the door, of course, for gene flow to occur through cross-pollination. So gene flow has been widely studied on cultivated species between their wild relatives, but information on this in Cyclopia specifically is very limited. And of course, gene flow can cause hybridization, which will have consequences for biodiversity, as well as the genetic distinctiveness between Cyclopia populations. So the aim of my study is to determine the risk of cross-pollination and thus loss in genetic diversity between wild and cultivated Cyclopia species and populations through pollen contamination. And I have three objectives of my study. And these are firstly to determine pollinators of cultivated Cyclopia species, and then to determine the potential for cross-pollination between planted and wild Cyclopia, and then lastly to determine whether viable hybrid populations of Cyclopia can result due to cross-pollination between planted and wild Cyclopia of the same and different species. So for this presentation, I'll just focus on the first two objectives and the results that I've got so far from those. And then I'll also talk about some of my results from my hand pollinations. So the study sites were chosen according to a few factors. So firstly, of course, the honeybush farmers needed to be willing to allow us to conduct the study on their property or their farm. And then the honeybush farm needed to be in close proximity to wild populations of honeybush for the gene flow aspect of the study. And then the farmers also needed to be willing to not cut the honeybush flowers in terms of their harvest requirements. And this is, of course, because I needed the flowers for the pollination aspects of the study. So we found two suitable study sites. And the first one is in Pearly Beach in the Overberg, and the second one in Tweede in Joubertina. So the study species were chosen according to their importance in cultivation. And of course, after choosing the study sites, the species under cultivation in those study sites were then chosen as a study species, as well as the wild occurring species in that area. So the first species in Pearly Beach was Cyclopia genistoides, or Kistia, and it was both cultivated in Pearly Beach as well as occurring wild or naturally occurring. And then in Tweede there was cultivated Cyclopia maculata, or needle-leaf honeybush tea, Cyclopia subtonata, both cultivated and naturally occurring, and then also naturally occurring or wild Cyclopia intermedia or Bergtier. So I made use of floral observation plots to determine whether carpenter bees were the only pollinators of Cyclopia. And this is because I did a literature review beforehand. Carpenter bees are considered to be the pollinators of Cyclopia because of the floral morphology of the genus. So we did around 3,070 minutes of observations, which comes up to around 51 hours. And in that time, we identified 6 carpenter bee species. So these were Xylocopa capitata, Flavarufa, Rufitarsus, Caphra, Cyoensis, and Seychelles. And then I also supplemented my observation plots with camera traps to increase observation hours. And I also caught and swabbed pollinators for pollen, for the presence of pollen. I'm still busy analyzing the data from these, so I will just focus this presentation on the data collected from floral observation plots. So from the floral observation plots, I was able to determine a visitation rate of carpenter bee visitors. And I've divided the table into wild species of honeybush and then cultivated species of honeybush, and then the visitation rates of each carpenter species to each of the honeybush species. And you can see that generally both wild and cultivated species share similar pollinators, give or take a few here or there. And the same can be said for each honeybush species. It's generally, they share the same pollinators. And also you can see that the visitation rates are extremely low. Apparently this is common for pea plants. I did read a paper by Grobler and Campbell, who in 2020, and they quantified the visitation rate of carpenter bees to Cyclopia pubescens, and they also had a very, very low visitation rate. Their median was 0.0116, so that made me feel a little bit better. But although the visitation rates are low, it's still very clear that you can see that carpenter bees are the pollinators of honeybush species, and that's what we were going for. And then also, you can see here that Xylocopa capitata and Xylocopa caphra are the two, they have the highest visitation rate for each species. So you can see that they're very important pollinators in terms of honeybush pollination. So now that we know what are pollinating honeybush, it's important to determine whether these pollinators have the potential to carry pollen between cultivated and wild populations of honeybush. And to do this, I used a mark release recapture method of tracking. So what we did was we captured carpenter bees in the cultivated site, as well as in the wild, and we marked them with different colors of nail polish. And then we released them and went about our reobservation methods. And we had two separate sites of release, obviously for the cultivated and the wild site. In the cultivated, we marked the bees captured there in blue, purple, and white nail polish. And then in the wild site, we marked the bees orange and red. So in the cultivated site, we marked 103 bees in total. And in the wild, we marked 29 bees. And although this may seem like a very skewed number, this is simply because in the cultivated site, there's a huge abundance of forage available. So there's loads of bees at any given time, and they're very easy to capture and mark, whereas it takes a little bit longer in the wild site, but we did put the same amount of effort into each. So we had 85 re-observations in total, and most of them were re-observed in their sites of release. So where they were marked, they were re-observed in. So just to quickly familiarize you with the map. So this area over here, which is the blue, purple, and orange polygon area, this is the cultivated site. And just below it is the site of release. So when we captured bees here in the cultivated site, we would mark them and release them over here. And then the white diamond at the bottom is the release site for the wild site. So 72 of the 85 re-observations occurred here in the cultivated site. And for the cultivated, caught, and marked bees, they were all re-observed in the cultivated site, besides for one, which is this one blue dot at the bottom. This was a light blue marked carpenter bee that was seen 730 meters away from the site of release, which was pretty interesting. And for the wild caught bees, many of them were re-observed in the wild again, which are represented by these red and orange dots, but also almost half of them were re-observed in the cultivated site. So it's very clear to see that there's definitely movement of these carpenter bees between the cultivated and the wild sites. And you can see that there's huge potential for pollen transfer. So in order to find the actual foraging routes of carpenter bees, we decided to use radio transmitters. And we made use of the LB2X radio transmitters from Hollow Hill. They are the smallest and most lightweight radio transmitters on the market at the moment. They are just under 0.3 grams, and they have a 12-day lifespan, and they have a 14-centimeter antenna. So after attaching the radio transmitters to the bees, we track them using radio telemetry. So we use the TRX48 receiver from Wildlife Materials. And during tracking, we took GPS points, and then I eventually mapped the points to form polygons. I just want to note here that we only used one Carpenter Bee species to radio tag, and that was Xylocopa capitata, as you can see in the pictures. And this is just because we saw that Xylocopa capitata is one of the most important pollinator species from that visitation rate table. And also it's one of the biggest Carpenter Bee species as well. So it was able to fly and forage, which you can see in those pictures, while it had the radio transmitter attached to it. So, like I said, we took GPS points of while we followed each of the tagged bees' route, and then I mapped them. And this was important to do because this can really help in determining whether or not these bees are moving and foraging in between the cultivated and the wild sites. So we attempted to radio tag 9 bees in total. We attempted many more, but we got the tag onto 9 bees, but only 5 were successful out of those 9. So the other 4, after we attached the radio transmitter, they flew out of signal for the receiver, and we didn't see them again, unfortunately. Here's a zoomed in version of the map. So each of the polygons, they have different dashed colors or solid lines that represents each of the bees' route. And the red square, or the red diamond, I should say, at the bottom was the release site. The maximum distance traveled by any of the carpenter bees was 283 meters from the release site, which is up here. And the average maximum distance that was traveled by all of the carpenter bees was 145 meters, so pretty far. And just to put that in context, the closest wild Cyclopia subtenata plant is only around roughly 20 meters away from the cultivated site. It's actually just in this riverbed below the cultivated site over here. So although I didn't track as many bees, or nearly as many bees as I would have liked to, I do think that this information gives a good idea of the capabilities of carpenter bees to initiate gene flow between the cultivated and the wild honeybush species. So here are some of the results from my hand pollination crossings. And this table shows the pod and seed set percentages for each of the wild study species for each cross. And the crosses I did were within species and between species. For the between species cross, I used cultivated maculata pollen and cultivated Cyclopia subtenata pollen. So you can see here that we didn't see any autonomous selfing at all for either of the species. And then interestingly, the within cross results were that only between 3 to 4% of flowers produced pods, which was very interesting. And then the seeds that were formed from those crosses, only 50% for subtenata, 33% for intermedia, and none for Genastroides, which was also very interesting. Selfing, only Cyclopia subtenata showed some form of selfing. And then this is what we were really looking for, is the between species cross, we got 10 seeds in total, 10 hybrid seeds, and these were 4 of the subtonata and maculata cross, and then 6 of the Genostoides and subtonata cross. So we are planning on planting these seeds, and then we're probably going to give them to the Agricultural Research Council to do further genetic testing on them. So the important message here is that we know what the main pollinators of honeybush are. They're carpenter bees, and we can see clearly that they are moving between wild and cultivated honeybush. And we know that hybrids can form. And although the results for the hybrids were very low, only 3 pods actually formed, it's still possible. We've proven that it's possible that it can occur. And there's really a need for a planting protocol for honeybush farmers to prevent cross-pollination from occurring and from hybrids escaping into the wild. And this year, this is something that I'll focus on trying to achieve as kind of like a decision tree. So I'd just like to mention the funders of the project. So the project is funded by the South African Honeybush Tea Association. They receive funding from the Alternative Crop Fund, which is managed by Hawkgrow. And then also to my field assistants, who are Darren Bonner and Fabi Home, thank you. And of course to Genevieve Turon for helping me with data collection. And finally, last but not least, a very big thank you to the farmers, Peter and Erna Norris, Dianya Bad, and Johan and Almarie Kutzinger. Thank you.
Thank you very much, Kirsten, for that presentation. I must say I like the live presentations. I've been to some conferences where everything is pretty, but this is just so much nicer. Okay, so let's have a look at the questions. I'll just start from the top here. Question from Sam. How long were the antennae of the tags? 1.4 or 14 centimeters?
Yeah, it sounds like it's quite long, but 14 centimeters, they're very long.
Yeah, but the bees can still fly with them, and it doesn't seem to hinder them. So
No, yeah.
As I said, you took the largest species of bee. From Glynis, okay, that's just a comment. Thanks for that, Glynis. And another comment, and then there's a question. Any theories about why you found hybrid seed between only certain species pairs? So why only hybrid between certain species and not others?
So I'm going to steal this theory from Cecilia from ARC, but she truly believes that these hybrids are only formed between species that share the same chromosome number, or at least in the same range of chromosome numbers. And yeah, that's why I believe that those certain species developed seeds and pods.
Okay, I see a thumbs up. Yes, so probably from Anina. Yeah, so it's to do with the poly level. Spot on. Mm-hmm. Question from Sandy. It's just a comment. Always fascinating to see the results of tagged studies. Well done. But yeah, they are quite difficult to do, and you don't always get what you what you intend to. But it's great to do these. I think we need to move on, Kirsten, due to the sake of time. Thanks for that. For the lovely presentation.
Cool,
and
thank you.
Then we move over to restoration. So another very applied project, pollination and restoration. Lundy, can you switch on your mic and camera? Just show us you there. Lundy?
Can you hear
Is Lundy
me?
in the room? Oh, there we go. Okay, great. Yes. Okay, are you managing? Not yet. Not yet.
Yeah.
There was a photo of you that popped up, but I can't see your screen yet. So there at the bottom, There's this. Oh, there we go. Okay. Great. I can see your screen and I can
Okay.
hear you. So it's over to you, Landi.
Okay, great. And you can hear me. Shirk, are you there?
We can hear you and we can see your slides. Landi, it seems like there's a bit of delay on the line and it seems like it could be mine, but I suspect it's yours because mine has been behaving up till now. Maybe switch off your camera just for bandwidth.
I seem to have lost connection from Shirk. If they see me. Oh, Shirk, are you?
I am here. Landi, I suggest switch off your camera
Okay.
for bandwidth because it's a bit delayed your side. And then you can continue. We can hear you and we can see your screen.
I'm just trying. There we go. All right, can you see and hear me now?
Yes, we can, Landi. You can go ahead. Landi,
Can you see and
seems
hear
like
me?
we have got issues. Yes, we could. You can just go ahead, Landi. We can see your screen. Just put it in presentation mode and we can see and we can hear you. You can go ahead.
All right, so for my master's project, I tried to determine if an active plant-focused restoration approach restores pollinator visitation to flowering plants. So even though the importance of also restoring pollinators have been emphasized, there's still relatively few studies that integrate the results of vegetation restoration with pollination, especially in the fynbos biome in South Africa. So while the long-term survival of plants in restoration sites and the success of restoration projects is dependent on pollination, this might not automatically always be reinstated when plant species. And the degradation of land usually leads to reduced floral abundance, which affects pollinators, and the connectivity of restoration sites to other natural areas also influences their dispersal and their colonization. Then thirdly, plants with specialized pollination systems are more at risk of extinction due to only having one or relatively few pollinators. And then on the other hand, our generalist plants are less at risk. They use a wide range of resources. However, we need to test this in the restoration context. So the aims of our project was to determine if pollinator visitation frequency and species richness in a recently restored and fragmented site is the same as that of an intact natural site, and also if actively restored plant species are optimally or suboptimally pollinated. Thirdly, we also looked at the studied plant species to determine if they are generalists or specialists in terms of pollinator visitors, and if they are capable of autonomy at all. This is a map of our study site, which was located in the fynbos biome. It's in the City of Cape Town boundary, and it's indicated by the little arrow on the map. The restoration site involved in this study consists of critically endangered Cape Flats sun fynbos. So this is a historical photo of the area taken around now. And this area in the middle here is surrounded by the Mountain Racecourse horse racing track, and on the inside is the Cape Flats sun fynbos. This Mountain Racecourse section forms part of the Table Bay Nature Reserve, which is located on the right-hand side here, which is a larger City of Cape Town Nature Reserve. This is a photograph of the area today. You can see that the entire Mountain Racecourse has been built up around and completely fragmented from Table Bay Nature Reserve here on the right-hand side. So we had 3 study sites, of which the first was the restored fragment. It was this little triangular shape here that was restored in 2018. And then adjacent to that, we had the Nature Reserve fragment, which is basically refers to the rest of Mountain Racecourse over here. That's been actively managed as a nature reserve since the early 2000s. And then these two both form part of the greater Table Bay Nature Reserve on the right-hand side, which we refer to as the Nature Reserve Intact. So we chose 7 focal plant species that occurred in all 3 sites or were easy to introduce at the sites, and then we collected data between the 3 sites on pollinator visitation frequency, so number of visits per flower per hour, and then also pollinator species richness. Thirdly, we also collected data on seed set, and for this, as we saw in previous presentations, we also did the 3 experiments where we left certain flowers open to be pollinated naturally, and then did hand supplementations of pollen on a second group of flowers, as well as the exclusion bags that you can see on the photo there, which we put up to see if plants are autonomous and if they form seeds in the absence of pollinators. These are the 7 species that we focused on. At the top 3 turned out to be our specialists, and the bottom 4 turned out to be our generalists. So basically we recorded any pollinators that visited and identified them up to family level, after which we classified them into functional groups. So if a plant species was pollinated by 3 or more functional groups, we would classify it as a generalist. And if it was pollinated by less than 3 functional groups, we would classify it as a specialist. So the top 3 specialists here is Babiana tubiflora, a species known to be pollinated by 1 species of long-tongued fly. And then Brunsvigia orientalis in the middle, known to be pollinated by malachite and double-collared sunbirds. And then top right, we've got Watsonia mariana, which is pollinated only by malachite sunbirds. Then at the bottom, in the bottom left-hand corner, we've got, the bottom is our generalist species. So the bottom left-hand corner is Ferraria crispa, which is mainly pollinated by several fly species, as well as bees to a lesser extent. And then Moraia flaccida, Ornithogalum thursoides, and Trachyandra divaricata, all of these 3 are primarily pollinated by bees. Then our results. In terms of visitation to specialist plant species, we did not observe any visits on any of our specialist plant species during observation sessions. We didn't observe any pollinators, but we did collect data on seed set. So just here on the bottom axis, we've got our 3 sites. Indicate those 3 treatments: natural, pollen supplemented, and autonomous, that we tested for seed set. So Babiana tubiflora in the top left corner, there was no significant difference between natural and autonomous seed set, except in the nature reserve fragment in the middle by 55%. And there was also no significant difference between natural seed set and seed set by pollen supplementation, indicating to us that this species is doing well. It's being pollinated optimally, but it can also self-pollinate well. But it's doing well in the absence or perceived absence of its only pollinator. And then Watsonia mariana at the top right. For this, pollen supplementations or seed set by pollen supplementation was significantly higher than natural seed set in all the sites, indicating to us that it's suboptimally pollinated. But interestingly, we just had a very high natural and autonomous seed set at the nature reserve fragment in the middle by 93%. But still, even though autonomous seed set here was high, pollination was still not optimal. And then at the bottom, we've got Brunsvigia orientalis, for which we had seed set by pollen supplementations also significantly higher than natural at 96%. So the species is then also suboptimally pollinated. And you can see that we had no seeds forming in the bags, so it cannot self-pollinate according to these results. Then with regards to our generalist plant species, I'll start off with their visitation and species richness. So Ferraria crispa was the odd one out in the sense that it had a higher visitation frequency and species richness in the fragmented site. So pollinator visitation was significantly higher in the nature reserve fragment, and species richness here at the bottom right was significantly higher in the restored fragment, opposed to the nature reserve intact. That was at 71% for the visitation frequency and 55% for the species richness. This was in contrast to Moraia flaccida and Ornithogalum thursoides, which had a higher visitation rate in the intact site and also a higher pollinator species richness in the intact site at 71 to 100%. Then Trachyandra divaricata, there was no significant differences in visits or richness, which makes sense. The species had Apis mellifera accounting for 97% of its pollinator visits. So very much a generalist and doing well everywhere in all our sites. Then in terms of seed set, for the generalists, we've got Ferraria crispa and Trachyandra divaricata on the left. So these two, the autonomous seed set was significantly lower for both of them at 79%. However, still no significant differences between the different seed sets for the different sites for either one of them. Then Morea flaccida up here on the right. The autonomous seed set was good, and there was also no significant difference between natural seed set and seed set by pollen supplementation. So this is being pollinated optimally, but it can self well. Then Ornithogalum thursoides down here, very low autonomous seed set, but still pollinated optimally at all three sites. As you can see, there's no significant differences between natural and pollen supplemented. So our results were quite varied between the different species. I will try and summarize it. In terms of specialist plant species, connectivity of the restoration site proved to be an important requirement for pollination visits and subsequent seed set to take place, unless the plant was capable of autonomy, as we saw in the case of Babiana tubiflora. Babiana was also the only specialist that we looked at that was still optimally pollinated in any of the sites. Exceptions, however, was that very high natural and autonomous seed set of Watsonia mariana at the one site, the nature reserve fragment. We believe that that might have been due to favorable environmental factors or characteristics that's unique to the population at that site, but this will need to be investigated. Then in terms of generalist plants, connectivity also proved to be an important requirement in terms of pollinator visits and richness for 2 of the 4 tested generalist species. However, both these species were also optimally pollinated at all sites based on their seed set. And then there was another exception among the generalist plants, which was Ferraria crispa. So in contrast to expectation, the species richness and visitation of pollinators was very high at the 2 fragmented sites, the restored fragment and the nature reserve fragment, which we believe might have been due to the surrounding land use. These 2 areas close to high density suburban apartments, as well as the horse racing stables where there are lots of horses and lots of horse manure. And families like Californidae and Sarcophagidae would be feeding on that. And these flowers, of course, are in close proximity to that area. In conclusion, successful restoration of pollination showed to be dependent on the pollination syndromes of plants, the connectivity of areas, whether plants were capable of autonomy or not, and then to a lesser extent, the adjacent land use. Overall, the choice of plant species to include in restoration should also be based on the characteristics of the restoration site and whether the dispersal and foraging requirements of those targeted pollinators can be met. And also keeping in mind if plants are autonomous or if they can self-pollinate or not. And then although the introduction of our generous plant species showed to have a greater ability to facilitate ecosystem recovery, providing those sustained nectar sources and stepping stones for specialist pollinators in Cape Fynbos is important for ecosystem function, especially in our small urban nature reserves. Future studies should also aim to incorporate more long-term data collection, as 2 years is a relatively short time to assess ecological functions, such as pollination. And the viability of seeds that was formed through autonomy should also be tested to see if those seeds actually germinate. And then lastly, I would just like to thank the Cape Peninsula University of Technology for funding towards the project, as well as my supervisor, Prof. Geertz, and the involved students that assisted with data collection. I would also like to thank the City of Cape Town for their funding towards the project and continuously providing resources and specialist input and staff. Thank you.
Landi, for that presentation. Nice to see application of a lot of the pollination work we do and apply that to actual restoration happening in the City of Cape Town. So thanks for for sharing that. Landi, let's try and switch on your camera again. Let's just see if that works. It's nice when you answer questions.
Okay, camera's turned on.
Okay,
There we go. Okay.
Okay, so see there's a question in Q&A. So I'll just read it for us again. Is a species capable of autonomous selfing or autogamy really considered successfully restored? Won't it lose genetic diversity over time?
Yeah, that is the issue on autonomic species. Is it really, how long would it last in future? That we don't know. So I guess we can't really say that it is successfully restored, but that would be different from species to species. When you think of species and that, then the differences come in if you look at.
Yeah, and just to add to that, I suppose it in that way it also guides restoration, because there's no need to restore the species in an urban nature reserve like this small urban nature reserve if the pollinators are not around and won't be around in future. Then you spend a lot of time and effort onto restoring something that can't produce seeds. So I think in that way it's applicable. Okay, let me quickly see any other questions in the chat or the Q&A. Lonnie, maybe if I may, of course I've been involved in this study, but we've only selected a certain number of specialists and a certain number of generalists. But just sort of off the cuff, most of those species that were restored, were they mostly generalist or were there more specialists that we didn't consider in this study?
Mostly generalists by far, I think. Restoration has a lot to do with when you do your species choices. It's got a lot to do with what seeds are available and what is going to propagate easily. And especially with these small critically endangered sites, your sources are often limited. So we would have liked to introduce more specialists, but often if you want to push the numbers up to make it successful and have lots of propagated material, lots of seed material, then you tend to stick more with the generalists. We should be in a place where we focus on specialists as well. But in terms of resources and what's available, you go for the generalists.
Yeah, pollination specialist, we will have to say. But yeah, it's a seed source, right? If they don't produce seeds, then you don't have a local seed source to restore. So in a way, they filter themselves. I think we have time for one last question. And it's a question from Anina. Do you expect the situation to improve for bird pollinated species over time? So more flowers. I suppose, over time after restoration, plants mature, they produce more flowers. So do you think then that will attract more birds?
Anina, I'm not sure about that, because with Brunsvigia, for example, we also didn't see visits in the intact sites, and there's lots of flowers there. There's lots of floral abundance there. So I think that we saw southern double-colored sunbird around, but I never saw malachite sunbirds around in the intact sites. So it depends on how far they can disperse. But I think it will be different from bird species to bird species. So maybe for southern double-colored sunbirds, there will be an increase. But malachite sunbirds, I don't know. I'm not sure what their dispersal distance is. These areas aren't so but yeah, I think the extent of urbanization in that study area has maybe just gone over the threshold that they can tolerate.
Yeah, thanks, Landi. Maybe for those that are interested in birds, there's a website recently launched on fynbos corridors, where all the stepping stones are mapped, and people are encouraged to add their stepping stones to it. And in that way, if you try and restore, you can see what are the best places to restore as to create stepping stones between urban nature reserves and with the larger nature reserves surrounding the city. Okay, so one more quick question, Nandi. Isn't the species with 79% visits by one species a specialist, even though the visitors are honeybees? That probably refers to your, I think it was Ferraria. Can you answer that one?
Can you that?
Isn't the species with 79%, so almost 100% visits by one species a specialist, even though the visitors are honeybees?
Well, that's a good question. Because we see the pollinator as a generalist, but then if the plant is only visited by one generalist, is the plant not a specialist? That's a good question. It kind of goes against our method, what we did. So Trachyandra divaricata, for example, we saw as a generalist plant, but it had a 97% visit from Apis mellifera, which we see as a generalist. So it kind of goes against our method of classifying them. But I think we must maybe just take into account that the pollinator is generally seen as a generalist.
Yes, Nandi. Yeah. So okay, but you also you said you classified the number of pollinator species or general family. I can't remember, pollinator groups. So even if it's mostly by honeybees, there were lots of other pollinators to those flowers, and that's why it was a generalist. Yeah. Anyways, I think we should wrap it up. We are over our time. I just want to thank all the speakers on this session, everyone did the presentations live, and lots of lovely photographs and figures, etc. So thank you very much. It's always nice to see what students come up with and the great projects they are taking on. And I want to thank all of you, the audience, for the questions, for the input. Really appreciate it. And I just want to use the opportunity to thank the organizers. I think it went very smoothly, and it was easy to press the right buttons. So just thanks for the organizers and for the technical guys behind the scenes in allowing us to run everything very smoothly. And thank you everyone. Enjoy the evening, and see you tomorrow morning. Cheers.